Yes, good afternoon, students. In the last class, we discussed regarding the entry of monosaccharides into the glycolytic pathway, that is, once after the formation of intermediate of glycolysis, such what we have observed like galactose, how it will have entry into the glycolysis, and as well as mannose and fructose, right? So now we will see other dietary disaccharides which are going to be hydrolyzed further to monosaccharides, and then it is going to uh, enter into the glycolysis, that is through various metabolic reactions. So how these particular disaccharides which cannot directly enter into the glycolytic pathway because it cannot enter the cells without first being hydrolyzed to monosaccharides, whereas extracellular that is in case of the invertebrates, this particular ingested disaccharides which is going to be first hydrolyzed by the enzyme attached to the outer surface of the epithelial cells lining the small intestine, then it is going to yield further monosaccharide units, right? So as such we can observe these examples like maltose, which is hydrolyzing by the help of enzyme maltase, and it gives you two glucose molecules, of course. So two glucose units will be formed, whereas in case of lactose it is going to be hydrolyzed and it is acted upon by the enzyme lactase, and it gives you galactose plus glucose, whereas sucrose, it is also undergoing hydrolysis in the presence of enzyme sucrase. It forms two monosaccharide units, that is d-fructose and d-glucose. Like such, as such, like we can also observe trehalose, it is also getting hydrolyzed in the presence of which is acted upon by the enzyme trehalase, which gives you two glucose units or two glucose moieties. So these monosaccharides which are going to be so formed are further transported in the cells lining the intestine. Thereby it is going to then bypass into the blood and are carried to the liver. Further they are going to be phosphorylated and funneled into the glycolytic sequences, that is, the glycolysis it is going to enter further and undergo various other reactions, that is, it follows the glycolytic pathway or the glycolytic sequence of the reaction. So we can see here in this slide you can make out that is lactose here and trehalose and as well as your galactose. So galactose it is uh, mainly it is going to be, I already explained you, it is going to react with the UDP to form UDP galactose, and UDP glucose will be formed further, and this UDP glucose which is going to get further converted into glucose 1 phosphate by releasing the UDP molecule. Then this glucose 1 phosphate further undergoing mutase reaction to form glucose 6 phosphate. Further, this glucose 6 phosphate is getting converted to fructose 6 phosphate. As such, this fructose 6 phosphate, as you all know, this glucose 6 phosphate itself can have entry into the glycolytic pathway, and it follows the further glycolytic sequence of the reactions. As such, fructose 6 phosphate and fructose 1 6 bis phosphate and glycerolidate 3 phosphate, right? So in this case of mannose, you can see here mannose which is undergoing exokinase reaction to form mannose 6 phosphate, which is catalyzed by the enzyme phosphogluco isomerase. Now you can see here mannose to fructose which is going to be formed, that is, those are the isomers, mannose to fructose. So it is acted upon by the isomerase enzyme to form fructose 6 phosphate. Further it continues the glycolytic pathway as such, which is involved in the sequence of glycolysis reactions. So we can see in case of lactose also it is acted upon by the enzyme lactase, and further gives you, as I already told you, lactose will be getting hydrolyzed to form your what is that, galactose, right? Galactose and glucose. So this glucose can undergo further exokinase reaction to form glucose 6 phosphate directly. Then it can continue the glycolysis pathway, right? Glucose is entering into the glycolysis directly, but galactose as such cannot enter. So it is formed by, it is undergoing sequence of reaction, then only it can have entry into the glycolysis. Whereas trehalose, it is acted upon by the enzyme trehalase. So you can see here two glucose moieties are going to be formed. So glucose it can have entry into the glycolysis as such directly. So you can see here further this another molecule. So you can see here sucrose, right, that is giving you glucose and fructose, right, two glucose units. So this glucose as such it enters, whereas fructose it has to undergo several enzymatic reactions. So it is acted upon by the enzyme fructokinase, wherever I am mentioning exokinase, fructokinase and all ATP is involved, it is undergoing phosphorylation reaction. Please remember This is very, very important. And fructose, without one more thing in metabolism, without cofactor, without coenzyme, without enzymes, none of the metabolic reactions are going to happen. So if you write without writing any cofactor, coenzyme and enzyme, definitely you will get 0 for that enzymatic reactions. You will not be getting even single mark for it. Whole complete metabolic pathway will be wrong. It is like a math understanding. So please go through properly each and individual reactions with the coenzymes, cofactors and mechanism why this enzyme is involved in this particular reaction. You have to understand what is the mechanism of that enzyme. Sorry. So here it is undergoing kinase reaction. So that's why ATP is involved. Phosphorylation reaction. It is phosphorylating at the first carbon atom of fructose to form fructose 1-phosphate. Then it is acted upon by the enzyme aldolase. Further, fructose 1 phosphate bisphosphate 1-phosphate aldolase. So you can see here aldolase is the enzyme mainly involved in the cleavage. We have already learnt in the glycolysis. Sorry. So in the glycolysis, so aldolase which cleaves the enzyme, cleaves the molecule, that is fructose 1-phosphate to here glycerolide, only glycerolide. There in glycolysis, glycerolide 3-phosphate plus DHAP. Why? Because fructose 1,6 bisphosphate over there. But here only fructose 1-phosphate, try to understand. So aldolase cleaves fructose 1-phosphate into glycerolide plus DHAP, that is dihydroxyacetone phosphate. This glycerolide is acted upon by the enzyme again kinase. So we require kinase because it has to undergo phosphorylation again. ATP is involved to form glycerolide 3-phosphate. Now this DHAP which is acted upon by the triose phosphate isomerase enzyme further getting converted to glycerolide 3-phosphate. Now glycerolide 3-phosphate as such it can enter into the glycolysis and continue the sequence of the reactions of glycolysis. Understanding? So as I have shown you here galactose very simple way in the last slide, so you can easily understand here. Galactokinase enzyme phosphorylation reaction to form galactose 1-phosphate. Then this UDP glucose galactose phosphate uridylyl transferase enzyme which is involved in the transfer of the this particular UDP group to galactose 1-phosphate to form glucose 1-phosphate plus UDP galactose, right? Understanding. So this glucose 1-phosphate and UDP galactose is going to be formed. So this UDP galactose is acted upon by the enzyme UDP glucose epimerase enzyme. So UDP galactose is getting epimerized to UDP glucose, just epimer. Glucose is an epimer. Galactose is an epimer of the fourth carbon atom of glucose. You might be knowing this. So this galactose is getting UDP galactose to UDP glucose, that is just an epimer. So this glucose is further UDP glucose, it is continuing again in the reaction sequence. Further UDP glucose is utilized for the reaction to carry out further. Again it is a repetition of reaction. So you can see in the last slide very like few reaction which you may not understand properly. So I have given you the complete sequence of the reactions of galactose. So have you understood? Now we will move on to the what is the intolerance. What do you mean by this lactose intolerance? It is a condition where in common where we can see this is very common in among the adults. In most of the human races we can accept the northern Europeans and as well as some Africans, which we can see this ingestion of the milk or other foods which contains the lactose, which further leads to abdominal cramps and even diarrhea. Abdominal cramps and even diarrhea because of could not able to digest that particular content that is the milk, right? Lactose intolerance is going to happen due to the disappearance it is going to disappear after childhood of most of all the lactase in the childhood of most of the childhood in the childhood days, but and even all of this lactase activity of this intestinal cells can be control. So in case of many childhood after this particular lactose intolerance is going to disappear, but this lactose cannot be completely digested here and absorbed. Whichever the patient whichever the individual who cannot digest this lactose completely, and it cannot be further absorbed through the intestine, of course. So this lactose which cannot be absorbed in the intestine, small intestine, which is further converted by the bacteria in the large intestine into the toxic product. So which as such it cannot be absorbed by the intestinal cells, small intestine. So it has to be further converted by the bacteria in the case of large intestine and converting it into toxic products further causes the symptoms of the condition. That's what is called as lactose intolerance. That is nothing but the individual who cannot able to digest the milk, that is ingestion of the milk in the foods which contains the lactose, whatever the dairy products or food, milk products which cannot be digested by the individual, which leads to further so many complications, that is abdominal cramps and even diarrhea, and even this particular converting into toxic products can happen. All these things which we can notice in case of the lactose intolerance. So in those parts of the world where lactose intolerance is prevalent, that is in case of most of the European, Northern European and some African countries, so milk is simply not used as a food by adults. Milk products digested with the lactase are commercially available in some countries. As an alternative to exclude these milk products from the diet, the only one option, that is they can exclude only the milk products from their diet. In certain diseases of the humans, especially several or all of the intestinal disaccharides which are going to be missing because of the genetic defects or the dietary factors, which further resulting in digestive disturbances, of course, and mainly triggered by disaccharides in the diet. So altering the particular diet to reduce the disaccharides content sometimes uh, elevates the symptoms of these defects, of course. So you have to control this particular disaccharide in the diet. Of course, you can you can avoid this particular, which further leads to sometimes elevates the symptoms of these defects, that is lactose intolerance defects also. So the defects in any of these three enzymes in this pathway, especially what we have discussed so far, that is galactokinase enzyme maybe, or UDP glucose galactosidyl transferase maybe. So this galactosemia, which we can see in case of the humans because of the galactokinase enzyme deficiency, because of this enzyme deficiency, this galactose cannot be further converted into uh, further it cannot be converted into glucose, so as such, so it further leads to high concentration of the galactose in the blood, so leads to galactosemia, which we can found in both in the case of the blood, of course in case of the urine also. So infants which develops the cataract, which is mainly caused by the deposition of this galactose metabolite, that is called as galacitol. So it is galactose is getting converted into galacitol, so further leads to the cataract formation in case of most of the infants in the lens. So this is the structure of galacitol. Please observe what is the difference in between galactose and galacitol structure. Just a small change in the structure, right? You can understand. That is aldehyde group is getting converted into alcoholic group, you can make out in the first carbon atom. So that is a galacitol structure. So symptoms of this disorder are relatively mild, especially, and very strict limitation of this galactose in the diet greatly diminishes this particular severity of the disease can be controlled. So transferase deficiency also can be seen. It is a more serious disease, and it is mainly characterized by the poor growth in case of the children. That is also galactosemia considered as. So speech abnormality is going to happen, mental deficiency, and even liver damage that may be fatal even when galactose is going to be withheld from the diet. Epimerase deficiency, galactosemia, also can lead to the similar symptoms, but it is very less severe than the other transferase deficiency galactosemia. So when dietary galactose is going to be carefully controlled, you can control this particular galactosemia condition in case of infants, and you can avoid the cataract formation as well as further severity of the condition, disease condition can be controlled. So this completes your entry of monosaccharides and disaccharides complete into the glycolytic pathway by the formation of the intermediate of glycolysis, and the disorders which are associated with this particular what is that pathways. And one more disease, that is fructose intolerance, same thing. So because of the enzyme deficiency fructose cannot be further converted into fructose 1 phosphate, so it is not possible to get phosphorylated at the first carbon atom of the fructose. So the fructose goes on getting accumulated in the blood, leads to the fructose um, intolerance, right? Fructose intolerance or fructosemia. So fructosuria, sorry, fructosuria and fructose intolerance. So you can see that fructosuria is nothing but increased concentration of the fructose in the urine. Of course, if it is going to be high level or the high level concentration in the blood, it leads to further in the urine we can see that excretion of the fructose content in the urine also, that leads to fructosuria. So please go through all these disorders associated with this metabolic pathway. Thank you.